We're all sharing like
All right. Good morning, everybody. It feels good to be standing up. All right. I think we left off right around here. I know we talked about hand washing. There's just a couple slides left to wrap up chapter 13, and then we're going to bump over into 14 here in just a second. So a couple extra molecules, chlorhexidine and alexidine, both of these antiseptics that we think of in commonly used in surgical scrubs, potentially like what we're seeing here. There are other agents that we think about that impact microbes by interacting with their nucleic acids. Alkylating agents get in and incorporate into these molecules, and we can see a couple of these right here. I mentioned somewhere in passing all of the plastic Petri dishes that we put all of the media in. They come in a sleeve. They are sterile in that sleeve, and they're sterile with ethylene oxide. So handfuls of different potentials here that get in and damage the nucleic acid, and in turn render whatever it is sterile. And these would be useful in situations like those Petri dishes that can't withstand autoclaving. So they tend to melt. That's not so good. You probably saw some of this activity in the lab a handful of weeks back when we did catalase tests. So many organisms have the enzyme catalase that gives them the opportunity to deal with harsh substances, stuff like peroxide, that some circumstances we might think of as maybe a cell producing this as an agent to combat something, and having the opportunity to have this enzyme gives them a leg up. So they basically, with this enzyme, take the harsh substance of peroxide and chew it up and turn it into water. So we generate water and oxygen gas. That's the bubbles that you see. And you likely saw a slide with lots of bubbles happening on there. It becomes important, not this we're seeing as cleansing stuff like some contact lenses. But when we think about it in the lab, catalase test is important for a small amount of microbes. When we think about trying to distinguish staphylococci from streptococci, a quick catalase test can clue you into some details. Boatloads of other bacteria also harbor catalase, numerous gram negatives. And doing a catalase test on those isn't truly very informative because you kind of just expect most of those are going to be positive. But when it comes to the staph and strep, they are not. And so it's an easy test. Staphs are positive for it, streps are negative. So this is a different circumstance. So this image, part of me thinks, why don't we show some of this in the next chapter? Because it truly kind of fits with that. But it also fits with all of the varied chemical agents that we're thinking about. I don't know when, but I assume in the very near future, you're going to do something like this. Have you already done this? Antibiotic, or not antibiotic, but different chemical stuff? Yes? Okay. And did you have little filter discs that you soaked in some stuff? Okay. So that's what we're looking at here. Those, not the same, but those filtered discs also could have antibiotics on them. And we have a load of different antibiotics on these little discs. Now you could dispense them onto a plate and do a same sort of an idea with a lawn of growth to see what sorts of antibiotics could be effective at treating these infections. And those plates, the real plates, remind me of that sort of stuff. And as I look at it, deep down, I think that's what we're really seeing here, because all of these discs look like they've got something printed on them. All of the antibiotics have some abbreviations on there. I think that's what this is. All right. That's why I'm like, this ought to be in the next chapter as well. So all of these could be different agents. I don't know what you tested, but I assume you had a plethora of different things that you could sample. Part of me, back in the day, and I don't think we did this, maybe you did, but sometimes students would bring in things from their house just out of curiosity, like, oh, I use this for cleaning. Is it effective? Or maybe I like these essential oils, whatever they may be, and I think they're super good. Are they effective? Maybe they are. Maybe they're not. Maybe they smell good, right? Maybe that's okay too. So can't tell you about any of that stuff, all right? Potentially, all right? And I assume with that battery of different things, you saw some differences. Some stuff maybe worked, some maybe worked a little bit, and some of it flat out smelled good, right? And that might be all it did, all right? So this zone of inhibition, the clearing on this plate, indicates whatever it was had some effect at killing these organisms or potentially slowing their growth. And in those instances where it slows, you might see sort of a halo and then some little speckles of bacteria that are growing after it's had a little bit of time to maybe dilute itself in that media, and they start growing. With antibiotics, sometimes you see that stuff, and we might classify those antibiotics as an intermediary, something that could slow it, but not truly kill it. And that would not be a drug of choice, right? You don't want to just slow them down. You want to annihilate them, all right? So you measure zones, and in this circumstance, those with the biggest zones of inhibition likely would be your best choice. Probably when you did this, you looked at some gram positives and gram negatives to make a comparison between the two. What's up with the difference in these types of bacteria? And that's what we got going on next door. So Pseudomonas aeruginosa, gram negative rod, Staphylococcus aureus, gram positive cocci, different, all right? I like the Pseudomonas. This isn't the best, but typically when Pseudomonas is spread out onto a plate, like a nutrient auger plate, and it's had a good couple of days to grow, the bacteria, as it's growing on this media, it produces some pigments, a greenish pigment called pyocyanin. And when we make these plates, we say we're making a lawn of growth. And when Pseudomonas turns green, I think, sweet, that's really a lawn, right? That looks like a nice green lawn, all right? You can kind of see a greenish tint here, but it's not as green as it may potentially get. So a couple of things here, and as we bump it over, like I said, antibiotics, there are definitely differences between antimicrobial drugs, some of them being broad spectrum, targeting both negatives and positives. Some of them are narrow spectrum, honing in on a specific type, a negative or a type of negative, potentially, or the same with the positives, all right? And maybe it could be in the patient's best interest to take a drug that is a narrow spectrum drug that's truly targeting that specific type of microbe, as opposed to a broad spectrum where you're wiping out lots of other bacteria in the process. That sets the stage for other problems, all right? So the zones, same idea as looking at these different chemical agents. We measure the zones, and ultimately each of these discs have a range where even a small zone looks like it might be doing it, may not really be your drug of choice. A great big zone, typically we think those likely would be a better drug of choice. But then you also have to think a little bit about patient history stuff. If penicillin is the drug of choice here, but the patient is allergic to penicillin, that's not going to be the drug you're prescribing, right? So you've got a couple of other opportunities where maybe this one isn't penicillin and they don't have allergic reactions to that. Similar zone size, probably our drug of choice in that circumstance. Looking next door at Pseudomonas, my gut says all four of these are the same antibiotics that we're looking at on these plates. And Pseudomonas, three of them don't do anything. They might be targeting gram positives only, all right, maybe. And I can see all four look like they're active with our staff. And then a little one up here with the Pseudomonas, a little bit of inhibition, nothing like we see with the staph, all right? There are some issues with Pseudomonas. There's issues with lots of bacteria that have resistance, all right? And resistance can be things that get acquired, horizontal transfer, or other opportunities for one microbe to share some resistance with some different microbe. And others just have it naturally. And Pseudomonas is one of those that is pretty resistant naturally to numerous antimicrobial agents. It sets the stage for issues, for sure, trying to treat Pseudomonas infections because we don't have a lot of drugs that are super good at treating. It also likes to form biofilms. It's got a very good capsule layer that makes it difficult to truly get those drugs in there to the organism itself, right? So lots of problems. And we'll talk some details of Pseudomonas, well, both of these guys, a little bit later. So I like this. I think this is interesting. We have never done any of this in the lab, and I think it could be interesting. Part of me wonders, is this benchtop disinfectant actually effective at its job? I'm assuming that was something that was put on a disc and you put it onto a plate. And I hope that it was effective, but I don't know. Anybody? Yes, was it effective? It wasn't? Okay. No zone of inhibition. And did you test a positive or a negative? Do you remember? A negative. E. coli. And it did nothing to E. coli. That's a little bit disturbing. Anybody else? Same deal? And did you also have E. coli or did you have somebody else? You used E. coli and nothing. Okay. Interesting. All right. Anybody with a gram positive? Were there any positives used in that stuff? It was all E. coli. Okay. Well, that's kind of an issue. So I think that's interesting. And maybe a bit disturbing. And you're like, well, what is this crap? Right? Are we just spraying water on the bench top? I don't know. That's a good one. All right. So this test, I think this is interesting. So here's our disinfectant, and they add some of the disinfectant to some broth that has I don't know what it's got in there, but it's got something that's supposed to inactivate the disinfectant. All right. So what we're really looking at here, we could have our squirt bottles that oftentimes don't squirt. I'm assuming you've got that going on. I don't know what's up with those, right? Uh, so you could take from that bottle, imagine this was the squirt bottle, and sample it just to see what's in my jug, right? Is it effective? Is it contaminated? Which is what we're really looking at. Did you say probably? Okay, so we take some of this, add it to the inactivator, and then we take from that tube and we put a little bit onto two different plates. Basically, two different incubation temperatures. One's going into the 37 and the other's just sitting out on the bench top. So we look and see, are there organisms that are ultimately growing, organisms that are in this stuff? All right. That's what we're looking for. Which makes me wonder, maybe we should get some of this inactivator just to see what's truly happening, or just streak right out of that disinfectant and see what's up, right? Little spritz on there if you could actually make it spritz. I bet you can't. All right. So last little bit. CRE is carbapreme resistant enterococcus. That's what we're looking at here, although these don't look like a coccus. So I don't know. So these guys, enterococcus used to be a streptococcus, and that's beside the point. These guys, these gram-positive cocci, become big concerns when we think about different things like some GI issues. They're super important when we think about individuals with diabetes and all sorts of foot issues and potentials for amputation. Staph infections, pretty common, all right? Staph infections, along with enterococcus infection, a co-infection, sets the stage for some serious issues. And these suckers are highly drug resistant. A co-infection with the two, kind of think an amputation is going to be happening. All right. It's bad news. All right. So these are common culprits in nosocomial infections or hospital-acquired infections. And some of these circumstances can be with different types of procedures where the devices may not be sterile, all right? Which, as I look at this, if I was this guy getting this duodenoscope, I'd be like, could I have a brand new one, right? I just bought this one. Could you just use this, right? Because it can't get ran through an autoclave. It can't get sterilized like we would hope all sorts of surgical equipment truly is being sterilized. All right. It gets cleansed. All right. But truly sterile? Probably not. All right. And this in turn sets the stage for some issues. All right. From one patient to another. All right. Disturbing. All right. Maybe it's not. Maybe you're like, it's fine. Okay, shifting over. Maybe come back to me. So this goes hand in hand really with what we were just thinking about with all sorts of different chemical agents. But now we want to think more about taking something into our body instead of maybe cleansing the bench top or the countertop, whatever, all right, or your skin, all right? So all sorts of different antimicrobial agents that we think about. Can I hide this? I know I can somewhere. Okay. So initially, penicillin, our first antimicrobial agent, we'll think a bit about Fleming here in a second. So first recognized early, mid 1920s, right around there, for its effect. Alexander Fleming was the one that recognized it. And then shortly thereafter, we ramp it up to thinking about broad applications and production, growing this organism in mass quantities to extract these drugs. And when it was first discovered, the thought, so many thoughts with discoveries that we think of as potentially a cure for everything. Penicillin was truly thought to be like this magic bullet, this thing that it will cure whatever ails you. A quick shot in the butt, forget about it. You're good to go. All right. And it was used for all sorts of things, not just bacteria. All right. And in turn, problems result shortly after we start using penicillin. It was highly effective against Staphylococcus, and few years, 1940s introduced. By the mid 40s, a lot of Staphylococcus already shows resistance against this. All right. Disturbing, alarming, all sorts of things, propaganda stuff that I think is pretty interesting. As we see here, saving soldiers' lives, they, speculate that, antibiotics truly did help the Allies because now we could treat. I think there were other helps with that as well. We don't need to go into all of this. But the one thing I truly enjoy as I look at some of these, I'm like, look at this guy chilling in the hospital bed, smoking a cigarette, just hanging out, right? All good. They were much different times back then, all right? Different, all right? So, and maybe that's truly what's helping him, right? I don't know, hard to say. So, we went full on with antibiotics and all sorts of research and development in all sorts of different agents that we could use to combat different bacteria. Because truly, when we're thinking antibiotics, that's what they're targeting. All right. So, overprescription, misuse, all sorts of things set the stage for resistance. It was really pretty commonplace for little kids to get some sort of a cold, to go to the doctor, and they would leave with a bottle of amoxicillin. Mom feels happy because she took you in and she left with something in her hand, and doggone it, she knows this is going to help you, right? But it's not. Many of these ailments were viral, and the antibiotics did nothing, all right? But potentially set the stage for some resistance, all right? So, problems, all right? There are also numerous instances where you are prescribed some sort of antibiotic, and typically these prescriptions are about 2 weeks worth of antibiotics. A week into it, you're feeling good. I'm better. Doggone it, I'm healed. And you quit taking them because why would I need to finish this bottle, right? I already feel better. And that sets the stage for issues as well, because the likelihood of you truly knocking all of those bad guys out, probably not. All right. And it sets the stage for problems. All right. And I know many people probably fall into that category and they're like, I don't take them all. Right. And maybe you still have some in that medicine cabinet just waiting. Right. Because maybe I'll get sick again. Right. Probably not wise. Eat those suckers. All right. So I wish I could look at this and spout off everything that I'm seeing up there, but I can't. All right. There are all sorts of different substances that we think about as holding some sort of utility. There are all sorts, as we see here, of different substances that Chinese herbalists have used for forever, all right? For millennia. We think about some of these things and wonder, what do they truly harbor, right? Medieval texts that describe weird concoctions to treat some sort of ailment. We're going back to some of these things and trying to see what's up with it. Can we replicate that? Can we find some sort of utility? You got to have a great big copper kettle, right? You got to cook it up. You got to do all of these things. That's what it said. Who knows what truly is lending to all of the activity, but they've been able to replicate some of those things. I think that's fascinating. All right. And like I said, I can't tell you what all of this is. It looks like a handful of different types of fungi. These look like something completely different. Looks like something that came out of the back end of an animal to me. But I don't think that's what it is. All right. Who knows? Could be medicine. And I can see we've got a chart here that's describing all of it, but it's probably in Chinese, and I couldn't tell you what it was in the first place. All right. So before we get to Fleming, I love all of these old photos, seeing individuals' labs and just all of the stuff. And it's my curiosity just runs wild. I'm like, what is going on? Awesome. So Ehrlich, he was studying syphilis, all right, and found a compound. I can't tell you. I should have done a little digging with this. Compound 606 is an antimicrobial that showed utility against syphilis. All right. And I assume 606 is probably like the 606th compound that he tested, but I don't know. All right. It's like WD-40, the 40th attempt, right? Then it worked, right? And then that makes me wonder, is 409 the cleaning stuff? Is it same deal? Was it the 409th, right? I don't know, right? Huh?
It is the 606th preparation.
It is.
It's
Okay.
Ersphenoamine.
Ersphenoamine. Okay.
That's so wrong.
Okay. That's all right. I would say it wrong too. So 606th attempt. All right. Perseverance. He keeps trying, right? And doggone it, nobody wants syphilis, right? We want to try to deal with this, right? So here's Fleming. 28, I said 1920s, late 1920s. So Alexander Fleming was the first to discover this activity, first to observe it, all right? He was studying Staphylococcus. And you can see in the background, we got a handful of Petri dishes back here. We got a lot of other stuff. The Petri dishes used back in the day, very different. They were all glass. You would pour your stuff. You could sterilize them in these dishes after it's been poured. Different, all right? The lids of these glass dishes fit rather loosely on the top. They don't seal right on there. The plastic Petri dishes don't seal, but they're a little tighter fit. All right. So there's all sorts of stories about this. And I don't know all of the details and what's truly accurate with this. But one of his plates that had Staph aureus on it. He had a handful of Staph aureus plates, but one of the plates, the lid wasn't completely on the Petri dish. It was a little bit off. So it allowed the air, it allowed microbes floating around to get onto that plate, and it allowed for them to grow, right, on these nutrients. So again, I don't know all the details. There's hints of an old sandwich. I don't know, right? Uh, Friday, then weekend. So a couple of extra days where nobody walked into the lab, numerous things potentially fitting into this perfect storm, if you will, this perfect opportunity for accidents to occur. All right. So he comes back to the lab after a few days and notices this plate with the lid that's partially off and sees some fungus growing on it. And the first thought is this plate's messed up, it's contaminated. You've probably seen a few Petri dishes in the lab that have a little something growing on them before you ever did anything. Contamination happens, right? It's hard to keep everything perfect, right? So he sees this, and I'm assuming the first thought is garbage, right? But he looks at it in the process of probably getting ready to discard it and makes a super important observation that where this fungus is growing, there is no staph growing. He can see staph on the other side of the plate, but just like that antibiotic disc I showed you, there's a great big zone of inhibition for that fungus. Something with it has made it so that the staph isn't growing. And he starts to wonder what's up. What's going on in this fungi that makes it inhibitory to the staphylococcus? All right. So he is the first to discover it. He makes these observations, discovers this fungus that has the capability of producing some sort of antimicrobial agent. Lots of different fungi can do this, and handfuls of bacteria can also do this, and they can secrete these substances into their environment. If you think of one of these microbes like a human, right, you might find some sort of food supply and you might like to keep all of that to yourself, and you might do something to try to protect it, right? Maybe these guys are doing the same sort of thing, secreting something to protect my food. And now others that might want to come in and eat my stuff can't. I'll kill them. And maybe I'll eat them too, right? All fine and good, right? That seems pretty dramatic, just trying to kill somebody to protect your food, right? But from a fungal or bacterial point of view, you do what you can, right? So he recognizes it. And our next two players, Flory and Chain, they're the ones that ultimately brought it to the market. So they figured out how to scale it up, growing mass quantities and extracting the drug from the fungus. All right. So they grew it up in mass quantities. They did antimicrobial testing. They did animal and human trials. And in the very early 1940s, right at the cusp of World War II, penicillin was released onto the market, thanks to all three of them. All right. And like I said, as soon as this happened, we're using it for all sorts of things. All right. Super common. All right. Another lab that I'm just like, oh man, look at all this stuff. What the hell's going on here? I think this is a still. I don't think he's making alcohol. All right. I think he is probably distilling something out of here, like maybe some of our antimicrobial stuff. I'm not sure. But a great big flask, a round bottom flask, getting heated up, and then right up in here, this would be the cooling chamber, and that tube going off into it looks like a smaller flask over here, kind of hard to see. So Waxman, once we start thinking about like I said, more than just fungi. Some bacteria can do this as well. Once we have this idea that there are organisms with the capabilities of producing these substances, all sorts of individuals got on board and wanted to try to find something. All right. You could find bacteria or fungi out in the environment. You could bring them into the lab and you could do similar stuff like those little discs that you had agents on. You could put some of the stuff you found on a plate that's got a lawn of growth and see, does it inhibit? There's all sorts of different opportunities, right? So he was working with some actinomycetes. These are a type of bacteria, broad groups of bacteria that live in the soil. And there are lots of different types of actinomycetes, numerous types harbor some sort of antimicrobial potentials, all right? And that's really what he put most of his life's work into, was understanding and identifying all sorts of different antibiotic producing actinomycetes, right? I recall as an undergrad in a microbiology course, so many things that I learned and sparked so much interest. But these actinomycetes, I was told soil smells like soil because of these guys. All right. This is what makes dirt smell like dirt. And I just thought dirt smells like dirt because it's dirt, right? That's why. You have probably smelled freshly tilled soil, or maybe kind of the smell after a rain. We are in Idaho. Maybe you've had the opportunity to be in a potato cellar where they are storing all of these potatoes. There is a smell that you can get in any of those instances, and that smell is dirt. It's really what it smells like. All right. But it smells like that because of these, right? You open those incubators in the lab, they don't smell good, right? They don't smell like dirt, right? I kind of think they smell like butt because most of the time E. coli is one of the dominant bacteria that you got growing in there. It smells like crap, right? Not so good, all right? But these, a little bit different, all right? A little bit better to smell, maybe, unless you hate the smell of dirt, then you're in trouble. All right. So this fits with a couple of different thoughts that I've already spouted off. It could be not taking your antibiotics properly, or it could be a situation of taking a broad spectrum antibiotic that sets the stage for some potential issues. And that's really what we've got up here, is the broad spectrum. So let's imagine that what we're thinking about here is your gut, all right? And we are taking maybe an antibiotic for something that's not gut related, all right? Maybe it's a skin issue, all right? And we're prescribed a broad spectrum antibiotic, and the hopes are that that drug is going to target this pathogen that we're really trying to target in the first place. But it targets other things as well, all right? So as we get to the gut, we've got a lot of organisms, buttloads of organisms that we can find, and a tremendous diversity of microbes that call that habitat home, all right? That's a great location. And some of the organisms that are in there have the potential to really be bad guys, all right? But there's only a few, because the rest of them have already taken up all of this space, and they maybe grow better. They're a little bit happier in this habitat, and they've taken it over. And so the pathogen, the potential pathogen, is kind of held in check by all of the other bacteria. All right. It's there, but not really showing itself like it could. All right. So in this instance, our broad spectrum drug, as it turns out, it kills everybody but our pathogen. Maybe that's true, maybe not. All right. But in this case, it is. All right. So we've wiped out all of these others, but for one reason or another, this guy isn't targeted by that antibiotic. It could have resistance normally to it. It could have picked up resistance from somebody else. Whatever the reason, it's got some drug resistance, and it's got resistance to that specific broad spectrum drug that we were treating it with. So now all of the real estate is opened up because all of the good guys got depleted. The bad guy replicates and proliferates and potentially causes serious issues. This is an example of Clostridium difficile. You have some of these anaerobic bacteria in your gut as normal flora. They're there, all right? They're not causing issues until you've wiped out the boatload of your other normal flora and lend to them proliferating. Clostridium difficile is difficult. It is very hard to treat. It requires some serious antibiotic therapy. And you were ill from something else. And now you're super ill from these guys. All right. And it can cause serious GI issues. We'll touch more on that. All right. So they're drug resistant in the first place. They didn't become resistant from this treatment. All right. It's not like this made them resistant. They already had the resistance. All right. And now they proliferate. And in this situation, we call this a super infection. All right. Where you have basically selected for these by inhibiting all of these others. All right. Lending to an overgrowth of this. There's other situations. We're talking bacteria right now, but we could slap Candida albicans into the mix. Also, some normal flora. These little yeast cells are in or on our bodies, but in pretty low levels. All right. And taking broad spectrum antibiotics sets the stage for those yeast cells that don't get touched by those drugs to be just like these blue pathogens right here. And you get an overgrowth of them, and then you wind up with yeast infections, all sorts of different locations in your mouth. We call it thrush. Same idea, all right, where you get an overgrowth, all right? So typically, trying to use narrow spectrum instead of broad spectrum helps to prevent these situations from occurring in the first place, not allowing the bulk of them to get wiped out. It's also, I feel like any antibiotic, but definitely with broad spectrum antibiotics, if those are what you're prescribed, you ought to think about some probiotics, yogurt or other probiotic foodstuffs. Consuming that sets the stage to minimize issues like this or yeast infections. It's definitely more of a concern with yeast infections, I think, but couldn't hurt. So when it comes to these drugs, there are numerous ways we may administer them. Oftentimes we think about oral. We take some one pill or two pills by mouth twice a day, typically is what's happening, but it could be an injection. It might be intramuscular, like a shot of penicillin, or it could be something a little more serious and directly put into the bloodstream, an IV administration. So looking at this, we've got a time frame down at the bottom. Could be days, whatever, doesn't matter. And then we've got the concentration of the drug going up the y-axis. We don't have any numbers here, all right? So the first one in red is the IV administration. And this makes sense. As you are first inoculated with that, injected with it, the amount of it that we can recognize in your plasma spikes dramatically. Shortly after it's been injected into you, you've got high levels of that drug in your circulation, spiking up, and then we see it declining. And this would be one shot, right? We're not talking repeated inoculations with it. It's just one shot, all right? One intramuscular or one pill, all right? So quickly spiking up, and then we see it starting to decline. Your body's metabolism starts to break these things down. As it starts to diffuse throughout the body, its utility also starts to decline, all right? The intramuscular, we see its levels increasing pretty quickly, but not spiking as much as we see directly into the bloodstream. It stays a little more pronounced. This is put right into the muscle, probably right near where we're trying to target that infection in the first place. And that in turn, not as high, but a little more prolonged with that before it starts to decline. And then in blue, the oral administration, it takes longer for it to start to peak here because it's got to get into the body, it's got to get digested and then into the circulation. It takes a little more time than either injection method, all right? And then it brings that level up and it also starts to decline, all right? I mentioned before, typically we think about like 2 weeks worth of stuff, all right? And if you're taking these pills, the idea is to get that level up and keep it at this level for a couple of weeks to have a high enough concentration to ensure that it's truly effective, all right? There are definitely problems with this. If you are taking your antibiotics like you should be taking them, you should kind of be on a schedule and you should take one in the morning and one in the evening, and it should be pretty close to the same time every time to keep it at basically that same level. If you miss a dose, it causes it to start to decline a bit, all right? Setting the stage for issues, all right? And if you take part of them and quit, we drop off, and the issues can go back to something like this, all right? Where, and I think there's another slide that's going to describe a little of that, but you start to knock out some of them. And in this case, let's forget about our pathogens and whatever. Maybe we could call all of these the bad guys. And you start to decline that population, but you don't get them all. You start to slow it down and kill some of them, but they're still around. And you quit taking that drug and they come back, all right? Because you never actually targeted all of them, all right? And that sets the stage. A few of those in that population may have some genes that render them resistant in the first place. And we've artificially selected for those populations to be enhanced. Going back and taking the rest of that drug, you still aren't going to have enough to combat it because now their populations have grown up. We only take half as what we should have because I already ate half the bottle, right? And you bring their population down a bit, but not truly where it should be. You go back to the doctor because you're still feeling like crap. They prescribed broad spectrum drugs in the first place. They didn't cultivate anything. They just say, take these will work. You show back up and they're like, geez, maybe we should prescribe something different because those obviously weren't effective. Wish I would have cultured it. Maybe they're wishing that. Maybe you're wishing that. And in turn, you leave with a different broad spectrum and you do the exact same thing. All right. Now you're setting yourself up for populations that truly might have some of these resistance, and you've enhanced it by the way you've taken those drugs. All right. Is that where I'm at? Yes. So this, I'm going to quit right with this. This is parental expectations of antibiotic use, 2000 and 2013, Medicaid versus commercial insurance, and what is the perception? I have some issues with this. I feel like it gives anybody that's taking Medicaid a bad name. It's like, oh, all they want's antibiotics, right? I think that's ridiculous. I truly question this study in the first place. You can see differences with all of these. The only thing that really looks like more of a decline is right here, and it's with both. All right, they have declined, but still misleading. One that I'm like, I should have deleted this before I even started lecture. All right, we'll quit with that. We will start off on Wednesday with different modes of action with these antibiotics. Good times. Thank you all. Good day.